Hello people, welcome to Smith Archives. Today's topic of discussion what we have is the rationale of endodontics and the fish zone of infection and its clinical significance. Okay, so Fish, he is a scientist who has been working on this, and he had given the zones of fish. There are 4 zones in the year 1934. Okay, so this is mainly based upon the reaction of the periradicular tissue to the bacteria, okay, bacterial products and its antigen, and the reaction of the bacteria to that particular antibodies, that necrotic tissue, okay, noxious necrosis products. Now there is a kind of experiment what that fish scientist he had did was, there was a pig, right? So what is that pig? Genoa pig. He took that lower mandible, and that he extracted the teeth, and then what he did was, in that space he immersed some cotton with the bacteria inoculation, and then he placed over the socket. Okay, why did he did that? He did that so to establish the foci of infection. मतलब उन्होंने क्या किया कि कॉटन लेके हम उसने जो भी बैक्टीरिया इनोकुलेशन था लैब में उसमें डिप करके जो भी कॉटन था उसने उसको सॉकेट के अंदर डाल दिया in order to establish the infection or the foci of localized infection. Okay, so this was किस लिए किया? In order to prove, or this was an early attempt to disprove the focal infection theory. Okay, focal infection theory को disprove करने के लिए इन्होंने क्या किया, कॉटन को डिप करते हुए बैक्टीरिया इनोकुलेशन में डिप करते हुए उसने जिनोवा पिग का जो टूथ का सॉकेट था वो भी मैंडिबल में इंसर्ट किया हुआ है. ठीक है, उसका रिजल्ट क्या आया? There was an infection. Yes, infection was there, but it was localized irrespective of the duration and its virulence. Okay, so this was all about the experiment which was done by Fish in the year 1934. Alright, so now we'll be studying about the 4 zones of the fish which he had given. Okay, the first one was the zone of infection, contamination, irritation and the stimulation. Alright, so we'll be discussing this detail. Okay, now we can see in the central zone. Okay, what is this? There is a bacterial inoculation first. Okay, so bacteria, what did it do? It does multiply. Okay, bacteria ज़्यादा से ज़्यादा multiply किया हुआ. ठीक है, so what does it produce? It produces exotoxins which are usually harmful to the tissue, right? So what these exotoxins did was, it degraded the host tissue and it forms the tissue degradation products. Okay, in the central zone we have 2 things, we have the exotoxins and the tissue degradation products, right? Now in order to respond to this, or with respect to this, PMNLs came into action. PMNLs आया अटैक किया बैक्टीरिया पे. ठीक है, now when this PMNL attacked the bacteria, exotoxins and the host tissue degradation products, they have migrated from the central zone to the periphery zone or the second zone, right? फर्स्ट जोन में क्या क्या देखने को मिला? पहले देखने को मिला बैक्टीरिया एंड पीएमएनएम्स दोनों ही यहाँ पे रहने लगा. ठीक है, सेंट्रल जोन बोलते हैं इसको, right? Also called as the zone of infection. Now coming to the second zone, which is also called as the zone of contamination. Here what we can see, existingly we can see something called as exotoxins and the tissue degradation products. Along with this we can also see something called as round cell infiltration. Alright, so this was also about the second zone or the zone of contamination. यहाँ पे कुछ नहीं होता, like just यहाँ पे जो पहले वाला central zone था, exotoxins और host tissue degradation products, TDPs, they just migrate into the second zone, which is called as the zone of contamination, and existingly there are presence of round cell infiltration, right? The third zone, which is called zone of irritation, यहाँ पे क्या क्या देखने को मिला था? देखो, this is a kind of a repair. Okay, यहाँ पे क्या क्या होने लगा था? फर्स्ट 2 जोन में डिग्रेडेशन देखने को मिला, पर थर्ड जोन में हमें रिपेयर देखने को मिला. सो व्हाट इज दैट? Dilution of toxins, that is osteoclast, osteoblast and macrophages, they come into picture here, and histologically we can see something called as a repair which is initiated by the body, right? हमें histologically there is a repair which is initiated by the body वो देखने को मिला था. ठीक है, there is a feel of heal here. And in the fourth zone what we can see is that there is mild toxins रिलीज किया हुआ बैक्टीरिया, सो दैट इज गोइंग टू एक्ट एज अ स्टिमुलेंट. सो उसके स्टिमुलेशन देखने को मिलता है किसको? फाइब्रोब्लास्ट और ऑस्टियोब्लास्ट को. ठीक है, फाइब्रोब्लास्ट और ऑस्टियोब्लास्ट दोनों स्टिमुलेट होके अपना अपना कोलेजन और हड्डी स्टार्ट बोन फॉर्मेशन स्टार्ट करता है, ठीक है. व्हाट इज दैट बोन? ऑस्टियोब्लास्ट, स्क्लेरोटिक बोन. ओके, so this is all about the four zones of the fish, that is four zones of infection, contamination, irritation and the stimulation, right? Now coming to the, now we have, this was all about the fish zone which was observed in the guinea pig, right? Now coming to the normal tooth anatomy of the human dentition, we have the same thing, right? We have the root canal which is containing the pulp, which it act as the site of infection. Now we have something called as microbes, right? Bacteria जाके inoculate करेंगे. Bacteria, they can't move, right? They have the ability to migrate, and they have the ability to migrate. They have the ability to increase their virulence, and they have the ability to increase their number and strength, right? So they are going to grow outside the canal. They increase in their number, they increase their virulence, and they're going to overwhelm the host defense, and they cause a periapical infection if they had this strength or ability to defend against the host defense, right? But if these microbes, if they get destroyed by PMNM cells, polymorpho-neutrophil leukocytes, they are going to form something called as a chronic pus, right? Chronic pus is nothing but it is the product which is produced by the bacteria when, देखो समझ लो कि बैक्टीरिया और पीएमएनएम का झगड़ा हो रहा है, राइट? इन दोनों का जो लॉस होता है, दोनों में हमें देखने को मिल जाता है. बैक्टीरिया का जो डिग्रेडेशन प्रोडक्ट्स क्या होता है, एक्सोटॉक्सिन्स होता है, और पीएमएनएम सेल्स का क्या क्या हमें देखने को मिलता है, डिग्रेडेशन प्रोडक्ट्स, प्रोटीओलिटिक एंजाइम से. इन दोनों के बीच में जो नेगेटिव पल्प था, होस्ट टिश्यू, वो भी डिग्रेड होता हुआ देखने को मिल जाता है, ठीक है. सो इसकी वजह से हमें क्या देखने को मिल जाता है, इंक्रीज अमाउंट ऑफ प्रोडक्शन ऑफ पस कंटेनिंग ऑल दिस थिंग्स, राइट? Exotoxins, necrotized pulp, along with proteolytic enzymes which are released by the dead PMNM cells, which all of this together constitute the pus. Which pus? Chronic pus. ठीक है. Now, the same thing which is explained in the periradicular area. In the last zone, what we see is the periphery toxins. They act as the stimulus and they form the granuloma. ठीक है. And the second zone, osteoblast and fibroblast, they stimulate and form the fibrous tissue, collagen. Collagen itself form the fibrous tissue, and the bone, which is formed by the osteoblast, which is the sclerotic bone. And the second zone, what we have is the bone of the area which is immediately vicinity to the root apex. They are going to be destroyed. Again, if the epithelial cells of molluscs, if they get stimulated by this thing, they get formed into cyst, right? So this is all about the fish zone of the zones of fish, right? And the rationale of endodontics. So here we have is the definition of rationale of endodontics. It is to remove the infected tissue from the root canal and seal it to prevent the reinfection. It also allows the surrounding tissue to heal, right? So this is all about the rationale of endodontics, okay? And the zones of fish. If you like the video, hit the subscribe button and like it. Thank you.